The overall goal of the following experiment is to induce and assess reprogramming of somatic cells towards pluripotency in their in vivo microenvironment. This is achieved by loading a syringe with plasmid DNAs encoding the Yamanaka factors and hydrodynamically injecting them into the tail vein of a Bussy mouse. As a second step, primary hepatocytes are isolated from liver tissues and relevant gene and protein expression levels are studied by real-time qRT-PCR and flow cytometry. Next, whole liver tissues are sectioned and stained by different techniques in order to confirm the reprogramming of the tissue and evaluate the toxicity of the procedure. Results are obtained that show differences in gene and protein expression of reprogramming pluripotency and hepatocyte-specific markers. Furthermore, the safety of the approach is also validated with biochemical and histological evidence.
The main advantage of reprogramming somatic cells in vivo is that it happens fast, efficiently, and transiently.
This method can help answer key questions in the reprogramming field, for example, the outcomes of inducing pluripotency in adult tissues in vivo.
To begin, anesthetize a 6-week-old female Bussy mouse. Then, prepare a plasmid DNA solution encoding the Yamanaka factors and a saline control as described in the accompanying text protocol. For the transfection to be successful, inject the entire dose in no more than 5 to 7 seconds.